Welcome back, everyone. It's January. We're doing our first conference call for the year 2024. We got lots to talk about. It's a very exciting time here at Foregen. I will begin just by going over a brief kind of summary of a few things that we've done these past few months. One has been fundraising. We are extremely happy and grateful for the support that you and everyone else in the community has shown when it comes to fundraising. As you know, we had a GoFundMe campaign that we launched to help shore up the funds that we needed for this second round of sheep trials, which, by the way, are going to be beginning in mid-February. I know that there was a bit of a delay with the ethics approval, and it's unfortunate. We wish it didn't happen, but these things sometimes happen. On the bright side, we do have the ethics approval, and they will be beginning, like I said, mid-February. So, but that being said, with the GoFundMe fundraiser, the community really pulled together, and we set a target for $45,000, and as of today, it's at $49,202. So that's really incredible, and has made it possible for us to continue our research in a big way. We also did a $50,000 matching campaign in December, as you know, and the community pulled through very well with that too. We raised $40,390. That matching campaign was supported by the Levine family, who, three of whom are on our board of directors, and they very generously put that together for us. And we're extremely happy that the community pulled through and helped us raise even more funds. And to be honest, the more that our war chest grows, the more that we can do, and the more options that we have, and the more opportunities we can pursue. So it puts us in a really good standing, and we're very happy with that. So on the research front, I'll just begin by talking a little bit about Histologics. So we get asked about Histologics a lot, and with good reason. As you know, Histologics we've contracted with, and they've been doing a great job so far on the study. We hit a snag in terms of sourcing tissue. As you know, we had the original source that we were working with to get tissue for that study was located in Ukraine, and when conflict broke out there, we couldn't rely on that source anymore, so we had to scramble to find another one. They're working with contacts on their end in the UK. They have two potential sources that they're working with to get tissue, and to make. We've also been exploring other options as well. So right now we're just kind of moving the ball forward on that. The good news is once Histologics does get the tissue, it won't be very long for them to finish that research, and it's an important precursor to ensuring that we're prepared for human clinical trials. When we do conduct those. So that's what I have for you guys. Right now, I'll pass it over to Enzo, and he's got more news and more updates to share with you. So, Enzo, when you're ready, go ahead and take the mic.
Okay. Thank you for your introduction, Ryan. Yeah, I just wanted to update you guys on what we did so far. The work in the cadaver lab is over. We have achieved a good result of surgery. We are imagining what kind of surgery we will perform on patients. We want to improve this surgery because, as you know, this is a multidisciplinary surgery. It involves not only a transplant of a human foreskin, but also linking small blood vessels. And also, we have to be very careful with the vascularization of the foreskin. So which involves, let's say, another specialist, and the fact that we want to regrow the nerve endings, which is our final goal, and also will involve other surgeons. So, I'm happy to tell you that we have, let's say, a kind of good idea of the surgery, as I said, and last week I flew to Düsseldorf to meet another great surgeon who wants to work with us. Probably somebody of you already knows him. Is Dr. Dan O'Day. He is specialized in reconstruct the women genitalia, the labia of the and the clitoris of a woman who has been circumcised. So he's an expert in this field, and he is very motivated also to work with us, because the work with us for him is the natural continuation of his work with women. So this is my main idea. We have conceived a general surgery in the cadaver lab, and now we want to perfect it with the specialist. And Dr. Dan Ode is one of them.
Great, thanks, Enzo. That's really exciting news. I know I'm really happy about that. William, do you have anything that you would like to talk about? Go ahead and grab the mic.
Okay, I can add a few extra details in there as well. As Enzo said, that it's a multidisciplinary, and the problem at hand involves more than just one or two doctors. So a big part of what Foregen's trying to do right now is make sure we have the ideal team to see this through and to make sure it's successful when we hit, not just through the clinical trial, but even before we hit the clinical trial. We're also looking at some other avenues of research, because we know one of the biggest issues right now is going to be the sample sourcing, right? We've already run into this. We're already dealing with it right now on our end. So we are aware of this problem, and we are talking with different research institutes about potential solutions that we would be investigating hopefully soon. Hopefully with some of these, we find a partnership and we create a research program that can help us be prepared for when the human clinical trial is done and there's a demand that can be met.
These past few months, we've been really spending a lot of time sort of networking and reaching out and probing for new partnerships that we could develop, because as William said, this really is going to require more than just a couple of specialists. So, and as Enzo alluded to as well, we're kind of piecing together a team of different experts to work on various aspects of this. As you may have seen in a previous newsletter, we've been talking to Dr. Jose Vega in Spain to help us to regenerate the myosin corpuscles. And of course, now we're talking with Dr. O'Day. He comes with a lot of experience. So there's also other partnerships that we're exploring right now. We can't talk too much about any details at the moment, but we're trying to make sure, like William was saying, that we're in a good place to make sure we can meet the challenge of scaling once we complete human clinical trials. That's an issue that's been raised many times by supporters, and we are very aware of that. We don't anticipate being able to treat tens of thousands of patients in the future relying solely on the sort of supply chain that we have right now. So yeah, I mean, we've got a lot of really exciting things that we're working on right now. Can't share too many details, but hopefully in the near future we can. Let's see. So right now we're basically prioritizing at this moment sheep trials. So those are beginning in mid-February. We want to get through those, of course, and conduct further experiments with the burying technique to ensure that the tissue is vascularized to the greatest extent possible before we attach it. Basically, with this kind of a surgery, the number one thing you want to avoid is tissue loss from lack of vascularization. So these sheep trials are very critical. We're also focused on getting the histology studies completed, consulting with outside experts, and building new partnerships. That's another thing that a lot of people have raised that question about building new partnerships, and we are focusing a lot on that right now. And we hope to have news to share with you in the future. As we've talked about a bit in the server, we are also working on hiring a research manager. We think that is going to be a key component of streamlining our research and increasing our efficiency and making sure that we can move forward more quickly and more effectively. And so we're prioritizing that right now as well. So once we finish these sort of four main areas that we're focusing on right now, we hope to position ourselves well to have a projected start date for human clinical trials that we can share with confidence. And that's where we're at right now. So Enzo and William, do you have anything else you want to add to that in terms of where we are right now and where we hope to be, by the end of this year? And then after that, we can open the floor for questions from the audience.
I can fill in a few minor details there from piggyback on what you've mentioned. I know, like we've mentioned, that we're looking for research. It's really a clinical research specialist, and it's not that we didn't have that direction. It's that it was several of us on the volunteer side all putting pieces of that in together. And we're getting to a point now where it's needed to get into the human clinical trials. That's actually a required piece of the human clinical trials. The as Enzo mentioned, we're talking with a doctor in Germany, so we're also broadening the horizon of the specialization, so to speak. We're making sure that we have the best doctors in the right fields to get the job done, and that's going to require quite a bit of logistical support on that end, and just communication support. We've always talked about that within the organization, that was a necessary component, and we finally hit that point that we felt that it really is. And that would also be someone working directly with the limb system, which we've talked about in the past. And we think we found the right limb system, but we want that person in place before we fully implement anything. And then, as Ryan and Enzo both said, with the surgeons and the outside consulting, you can think of right now as a surgical review. So one of the big pieces of the animal trial is we have to have a certain number of successful implantations into the animals that we're using to show that it's successful, that the procedure works for recellularization of a decellularized component tissue that's implanted. And once we have that, we need to apply that to the surgical procedure, the surgical plan. So right now, the other piece of that is we're reviewing that surgical plan with these other doctors and with these outside consultants. And if they see something, or if they have something to say about it, going back to the drawing board with surgeons and saying, hey, do we think this is a concern? Do we think that's more of a concern for this reason? Stuff like that. So maybe just to shed a little bit of light on a few things that you said there, Ryan.
Great. Okay. So anyone who wants to ask a question, go ahead and raise your hand, and we'll get you up here on stage.
You can also type them in the chat as well. I just wanted to remind everyone.
Will the surgical technique work on all types of circumcision?
Yeah, it should. That's one of the reasons why, like I mentioned, we're going back to surgical review, is to ask questions like this with different specialists. And as Enzo mentioned, he's not just a very passionate doctor about this very topic. He's very knowledgeable, and he's been doing it for women for quite a while, and he's very expert at it. So having those types of doctors on staff or part of this project is where we're hoping to, yes, answer that question. Ultimately, that would be consultation with the surgeon to see what you envision the outcome as, and for them to help you realize that. I hope that answers that thoroughly for you.
And to add a little more detail there, like when William says we're in a surgical review phase, basically what that means is the surgeon that we've been working with in Italy, as you've been following in the newsletters, has completed both of those 2 cadaver lab sessions and has delivered 2 reports on the results of those cadaver lab sessions to us. And now we are sort of assimilating that information and putting together a surgical plan. And we're inviting other consultants to review that information and provide input. So that we can, refine the technique as much as possible. So that's pretty much the stage that we're at right now.
What has been the reaction to Foregen from doctors and specialists, especially as you gain more and more credibility?
I think Enzo probably has a lot to say about that after meeting with Dr. O'Day, for example, or Dr. Jose Vega. Enzo, maybe you want to talk a little bit about their reactions to Foregen?
Yeah, I mean, it was a good reaction because the doctors I spoke with know incredibly to say very well the topic and know the problem. For instance, Dr. O'Day told me that women circumcision has been performed to 200,000 women all over the world. So it's a great number of women who have been, who received this surgery. And for instance, men, as you know, there are 1 billion of men, and the doctors know this. So Dr. O'Day knows this very well, and he has come up with some ideas to work with reverse the circumcision. So to answer also the previous question, obviously we are considering all the difficulties of the surgery, for instance, if the circumcision was too tight, if there isn't enough skin, if the frenulum is missing, how many Mesner corpuscles we need, and how many nerve endings we need inside the foreskin. And Dr. Ode was prepared. I mean, I saw him, in great. He was okay with this topic. He didn't have any problem. Actually, he knew a lot of things. So he is one of them. But maybe we can even add some other specialists to him. Let's see how we work together, because I think he has a good plan. The general meeting I had with him, he has a very good plan to go in the details of each different surgery for each specific patient.
What are your considerations for having the human clinical trials in a place other than Italy?
I can say that Italy has been our first choice for a while for a number of reasons. But, at this point, it hasn't really been settled on where we would do it. William, do you have anything you'd like to add to answering that question?
Yeah, so I know one of the big questions is, will the human clinical trial be performed in the U.S.? And where we're at right now, it looks like it's going to be in the EU. And the reason it looks like it's going to be in the EU is because the animal trial has already been completed there. And for us to restart that process in the US would set us back on the timeline, but also expense-wise. It would be fairly expensive to start that phase over again. Now, is that to say once it's completed, it wouldn't be available? No. Once it's completed with EU regulations, the ability to transfer that type of surgery over to US FDA regulations, it is usually much easier than just transfer something mid clinical trial. That's actually very tough to do in a lot of cases. So we're looking at completing it in the EU. That's where a lot of the surgeons and a lot of the doctors are. And in kind of the first part of that question, a lot of the doctors that Enzo's been talking to, that we've been talking to and working with, a lot of the ones that are more partial to what or understand our perspective and what we're going through and what we're trying to accomplish are in the EU. It seems like there's a fair amount of doctors in the US, but there's more in the EU that are actually actively working on something similar to this. Dr. O'Day, for example. So right now, that just seems like the best place to get this completed in a more efficient timeline and with the least amount of funds needed. Again, not to say it wouldn't be considered in the future, but that would also depend on what other partnerships we make and what other relationships we start. So it's open. We're not against it.
I just want to say that I think it's very important, I don't know if this is the right word, the climate. For instance, just by case, I found out that in Germany, there are many doctors who already work on reversing in some way the circumcision. So, we never found the same amount of scientists in the United States. And I think this has to do with the fact that United States is, I would say, a cutting country, and it's very difficult to go against this way to work. So this is the reason why we would like to do this human trial in Europe, because we see from the way the doctors work, they have more sensibility about this topic, while it's very difficult to find the same sensibility in reversing circumcision in the United States.
Thank you, Enzo.
How will the 25 patients be chosen for the human clinical trial? Is there any news regarding the LIMS software you were testing for this purpose?
Yeah, I think, William's been handling a lot of that in recent weeks. Do you have anything that you'd like to add, William, about the LIMS?
We've chosen the structure and the company we want to go with for monetary reasons and for efficiency and efficacy. It seems like it's very effective, and they're very partial to helping us out. Kind of at a hold off on that. We want to get the clinical project manager in place before we absolutely jump and commit to it. We also want to get the surgical team on board, or as much of them on board as possible, before committing to the structure of it too. The steps moving forward with that would be clinical project manager, then trickling in the surgical staff or getting the surgical team put together. That way we're not setting up a system that someone says, oh, it'd be a little bit faster, easier if I had this function in there. It's when I work with patients or something like that. Just from my experience using LIMS systems in laboratories designed by people who weren't ever meant to use the LIMS system, it's a great way to make something that people hate you for. So the point we're at, we found the company we like. We found the system we like that can be modified for our needs. We want to get the clinical project manager in place before committing to that system. And then we want to get the surgical team into place before finalizing the interface with that system.
During the clinical trial, how could Foregen assess the full regeneration for the nerve endings? Is it possible to compare that data to previous studies?
I mean, this is a very good question. And how can we compare the sensitivity of the man who never had the foreskin with the fact that now has a foreskin or will have a foreskin? And we have thought to that a lot.
Yeah, as Enzo said, we've talked about this a lot. And there's a few different methods of testing that. But Enzo said it's very difficult when you have someone who doesn't have anything to compare to. It's easier when you've had someone who was circumcised later in life and remembers, or fairly recently, and remembers the sensation, or remembers they have something to compare it to, is the bottom line. This is something that we've been discussing, and I think we'll come up with a better plan or a better grasp on how to analyze this as we get into the human clinical trial. Remember, that's also going to have effect on this. If you have a certain amount of time slotted for regrowth, and then a different type of surgery or different type of plan changes that timeline. So we're kind of struggling with finalizing and understanding that timeline right now. And then I think once we have that down, we can focus more on how we're going to analyze the progress with innervation. This is something that, as Enzo brought up, it's not just the surgeons. We've got neuroscientists and we have other specialists from different places in the world that we've been communicating with about this topic. And that's a big next step. That's a big piece of the research that we're going to be discussing next after the surgical review is completed and we have a clear picture of the surgical timeline. That I think that's a big piece that has to be answered before this question is answered fully.
There's been quite a lot of discussion over the last few months about connecting the frenulum to the glands. Are you able to say the plans you have for the procedure?
Yeah, I can answer this question. We are working with extracellular matrix. So, the goal is to have, let's say, enough foreskin from a cadaver. And in this case, we have only to transplant the matrix to the recipient in vivo. And in this case, the foreskin and the frenulum will be coming from a cadaver and will be attached to the recipient, to the penis of the recipient. And then we are going already, let's say that we will have a 70% vascularized foreskin and frenulum at that point. And so with our growth factors, we want to finish the vascularization, arrive to 100% in vivo. That's the general idea, the concept idea.
As the human clinical trials approach, has there been any talks about publicity or public education events?
Yeah, we have thought about that. I mean, I guess I could say off the bat, I think that we may be able to attract more media interest when we are closer to actually conducting the trials. I think that would be something that would grab a lot of attention when people see that it's actually happening. So I think that we could attract some media attention. Enzo, do you have any thoughts about that, about any public events or publicity surrounding human clinical trials?
Yeah, sure. I've always thought that before, when we have finished all our experiment with animals, with cadavers, and in the lab, and we feel 100% sure to face a human trial, we have to prepare that event in a certain way. So my original idea was to fly to United States and make a public conference in which you guys, you can participate, you can attend with all the data of what we have achieved so far. And at that point, also create a kind of interest and about to, some publicity about this upcoming human trial. Another idea could be to make a big, because art helps a lot. My experience tells me that the art helps a lot. It's not only something, I would say, exclusive, but, because it can reach a lot of hearts, a lot of minds, a lot of intelligent people, and also, could be an idea to do another UFO exhibition when, we are ready. This is just an idea that comes to my mind now. So we haven't thought to do this exhibition. It's an idea. But for sure, I would like to share with you in a public conference, the results of our work. Of course, the ones that will be possible to share before publication or just talk about them generally. And at that point, we will be ready. And so we will create the right momentum, the right, atmosphere for the human trial.
Will sourcing the extracellular matrices be easier for the human clinical trials than it is for the histology studies? Are you able to share details on that?
I would say, one thing that comes to mind is that the responsibilities for sourcing tissue with the histology study is not really with us, so that's sort of different from the sheep trials or the human clinical trials. So I can't say it would be easier, but probably. We're currently talking with a potential source in Europe for the future of our research that we may need as well. So I would say, yeah, probably easier. I think William and Enzo would know better than me and probably have more details about that. But one of the things about the histology studies, it's a complicated sort of web of responsibility on the histology study, and the war in Ukraine and COVID really delayed things. I mean, new policies, new restrictions, new regulations came into effect in terms of how tissue is transported during COVID, and I think some of those regulations are still in place, and that has made it more difficult to transport tissue. So yeah, I would say I think that it will be easier, but maybe William or Enzo, you have something to add to that about sourcing tissue for human clinical trials.
I can add that it has been difficult lately. It seems like it was getting a little bit better. As Ryan said, we definitely have a couple potential sources that are closer to where we're doing the animal trials right now. So that would actually be preferable, because if we need them for the human clinical, they're within a distance that transportation isn't ridiculous. As Ryan also mentioned, COVID regulations changed a lot of how things are transported, just made them more rigorous and more involved. That said, we will do what we have to do to get through the human clinical trial when we get to that point, and we'll make sure that we have everything ahead of time that's needed. But the difficulty of sourcing the samples, like we said before, was one of the reasons that we've jump-started and made it more of a priority to communicate with other research organizations, institutions, and see who is doing the research and where they're at, making engineered scaffolds and engineered materials, so that, ideally at one point we wouldn't have to rely on ECMs from decellularized cadaver tissue. Also, in that case, you potentially could make something to size needed. So we wouldn't have to, like we mentioned before, it'd be up to the surgeon and up to the Lynch system to pair the best sample, the best ECM with the patient that needs that ECM. And yes, there's a system that can help do that, but if the sample doesn't exist or we have to wait for the sample, that's all downtime that someone's waiting on the procedure. So yes, if we think it, especially outside of the histology study, it's going to be easier, but not seamlessly, not like 100%, like there's a surplus of samples for us. We're still going to have to do our due diligence. Enzo's been working really hard putting relationships together in Europe for these samples to be available for us for the human clinical. We anticipate doing this all now, making sure we have those sources, but there's still a lot of work to be done on our side.
It definitely became more difficult to find the sources. the first test we did, I found the source from United States who was finding the foreskins in Ukraine. I didn't know they were coming from Ukraine. But then at a certain point he said, you know what, I can give you the contacts, you can contact them directly. And that's what we did. We started this collaboration with this institution, and they gave the samples that we needed. But then it was more difficult for, the reasons that you already know. But, speaking with the other doctors who work in the transplant field, they say, this is normal for any organ. But what I did, obviously, to avoid any kind of other problems, just, relying on only one source, I'm looking for other sources, and I have found another one. So we have a meeting next week to understand, if they can provide the foreskins, the tissue that we need. So I just want to tell you something. When we don't communicate nothing, it's a good news, because it means that we are working, and that's the way I usually work. I don't say anything that before that happens, because I don't like to deceive people, and I don't like to say lies to people. So next week we are going to meet with this group of and they will tell us how many foreskins, how many samples they can provide. Because remember, we need a lot of tissues. For instance, if we can make a number, 25 people for the human trials, so we need at least 50 or 70 probably organs for that, to be sure. But then we have the histologic study, so we need hundreds of tissues. And so we work in different directions to find them. One of them, we have an appointment next week. Thank you.
Yeah, thank you, Enzo. And I just want to add on to that. Sometimes, like right now, it can seem a little quiet, like Enzo was saying, and it actually is a good thing. I mean, we've been doing a lot behind the scenes, and we've been really laying down tracks on some potential partnerships, like Enzo was just alluding to. So when it's quiet, we are working very hard behind the scenes, and we can't really update the community until after things have been, until after our work is paid off and after things have been confirmed. So of course we will let you know like how things play out when things can be confirmed. So hang tight, and we really appreciate your patience.
According to the information I read on the Discord, the last round of animal trials is scheduled to begin in mid-February and end approximately in mid-May. When are human trials expected to begin after that, assuming everything goes well with the last round of animal trials?
I'll just start and say that's a tough question. There are things we're focusing on right now, and until those are finished, it's difficult for us to have a clear picture on when we can do human clinical trials. So we just have to get through these main tasks we're working on right now that involves developing partnerships and consulting with outside surgeons on our procedure and finalizing which country we want to do the trials in and things like that. We have to do all these things first. So we're just being careful, because like Enzo said before, we don't want to raise people's hopes and then dash them. So we're just being very careful with timelines right now, and we will put out a timeline we believe we can hit when it comes to human clinical trials, but we just can't do that right now. We're going to continue to make progress step by step and keep you updated every step of the way, and we're working very hard behind the scenes on a lot of really important partnerships. So
something for instance, right now, imagine we have finished all the sheep trials, okay, and we have a lot of data, a lot of results from the cadaver lab, but we don't have the foreskins. Can you imagine? So how can we do that? So there are problems to find this tissue right now. So there are a lot of things that must be analyzed and resolved first, and we are doing this very well. You know how we are, we're people that want to do this trial. The sheep trials. They should start around February. I spoke with Angela Palumbo, February 10 or 12. So yes, mid-February. And we have the sheep, we have, this is important, we have the foreskins. So we are all set for the experiment. We have authorization from the ministry finally, as you know, and so yeah, it should last 3 months at least.
Yeah, that's the time frame. You never know if things have to be adjusted or if certain things have to be repeated, but it may be a little bit longer than that, but right now it's anticipated for it to last 3 months.
With organ transplants, the patient is often required to take immunosuppressants to prevent the body from rejecting the new organ. Will this be needed with Foregen's procedure?
No, in this procedure, we don't see that as being a necessary step because it's a decellularized. So the reason your body needs that transplant is because you don't have a full extracellular matrix. It still has the cells from the donor in it. So even if it's a family member, your body's trying to attack those cells. It's like putting ants from one colony into another colony. They don't like it. In this case, you're kind of removing all the ants from the ant colony, and you're just introducing new ants to that colony. And they're just going to go explore the tunnels and be like, oh, cool, this is home now. So that's, very simplified, but that's essentially what we're doing with an ECM. So the use of strong immunosuppressants is not something we're thinking is going to be necessary at all in this.
Will we have to take medication to suppress erections?
I think that the patients are going to take the same medicine they usually take when they receive their adult circumcisions to avoid an erection. And I'm not able to answer to this scientific question, actually.
I don't think we have an answer to this yet because the surgical plan hasn't been finalized. There's going to be some medicine, but I think that's also on a patient-to-patient basis. The doctor will prescribe the appropriate medicine for you. If you're allergic to one and there's an alternative, that would be something that you have to consult with your doctor.
Is bovine donor tissue still being considered for the procedure?
My main idea is to go in 2 directions. One is to resolve the problem with 25 patients in the human trial with extracellular matrix. But I'm working also in another direction, which is, for instance, if we want to solve this problem for 1 million of people, how can we do? Because there is a shortage of organ. Okay, we go in the direction of, the bioprinting. Yes, this is that.
I could simplify the answer there and piggyback off what Enzo said. We're not looking at bovine ECM as we're not looking at animal ECM as a purely decellularized animal tissue. We're not looking at that. We're looking at engineered ECM. And we're looking at as everything Enzo said. The reason we're going with engineered ECM is if we can guarantee acceptability by the body, and if we can guarantee innervation and vascularity, if we can hedge those bets and get something that's better than an ECM, we're absolutely going to try to do that. And as Enzo said, if we can manufacture it to scale for every individual that comes in to meet their needs. There was a question earlier about, will this procedure be available for every type of mutilation? And I think this is a big piece of that. In order to answer that, yes, we need a way to fill every need. So an engineered ECM, we're really thinking is the way to go. So the question that was asked, are we looking at bovine? No, some of the products out there are bovine derived, and that might be an option, especially in the interim, but we're not looking at bovine ECM. We're more leaning toward engineered ECMs.
For supporters located in the European Union, we all have unrestricted access to public hospitals and medical care. Do you think Foregen's procedure will be covered in the EU by governmental health policies?
To answer that partially, I don't think I don't have a solid answer for that. I think that might be dependent on each country in Europe, like how they want to handle that or how their system handles that. And if they consider it also depends on how they're going to consider the surgery. We've been looking at like the worst case scenario, you have to pay for it. Also, we're talking with different surgeons right now from different countries as well. You have to remember. So we're still kind of in that phase, reviewing that procedure to make sure the procedure works. And we haven't really gotten a chance to really sit down consider this topic. I think this will be something we really consider once we're successful. Once we get through the human clinical trial and we have a successful surgery that works, this is something that we can consider and actually go through the steps of communicating with different countries and saying, hey, how, can we, I think it's going to be lobbying is what I'm getting at. That's going to be some lobbying in those countries to say this is a necessary procedure. Otherwise, in the U.S., to answer your question about the U.S., I think it's going to be a paid for procedure. And unless a state somewhere says that it's, you never know in the U.S., the law can swing one way or the other, or somebody can lobby it and get it into a position where insurance will cover part of it. But I think in the U.S., it's going to be a much harder fight to do that than in the EU. There is a dogma, there is a certain social consideration or outlook in the U.S. that is very negatively toward this aspect of men's genitals, for just to speak frankly. Yeah, to answer that, I think it's going to be a while before this is something that the government in the U.S. even just considers paying for. And in Europe, it's something we haven't had time to consider yet. We've been really, Enzo's been working really hard. All of us have been working, communicating with these surgeons, communicating with these outside organizations, just considering the scientific and the surgical aspect. So, yeah, thank you for the question. I just don't think it's something we have the answers for right now.
Thank you all for coming, and I really enjoyed the discussion here, and we're really happy with the support that we've been getting lately, and we're very excited about the work we're doing, and we can't wait to share updates with you. Any parting words, Enzo, William?
Yeah, I just want to thank all the donors for their support. Without you guys, we couldn't be at the point where we are now. If I think at 13 years ago when everything started, I couldn't imagine we could arrive to this time, doing everything by ourselves without help, just raising the money personally, individually. You guys are great. Thank you.
I would also like to add, thank you everyone for your patience. Thank you for your support. I know there's times where we kind of go dark and we're just kind of furiously working here in the background, trying to get communication through to different parties and try to work some of these relationships that we've been talking about. And Joe mentioned that he went to visit some scientists and I went afterward to visit them and have further discussions more on the scientific side and planning side. There's a lot going on right now. Thank you for your patience. Really appreciate it. And thank you all for your support again and attending and the questions that you asked. They've all been really good. Keep it up and look forward to much more positive 2024 than a 2023. Seems like we're already making a heck of a lot more progress at the beginning of this year.
Yeah, 100%. This year's shaping up to be very different. So we really can't wait. Again, thank you all. And that's it for now. We'll see you in the next conference call. And just again, thank you for your patience. And let's see this project through. Let's get it done together. So thanks everyone.